Imagine you're tasked with building this incredibly vast, just sprawling plumbing system for a huge metropolis. You've got like unlimited materials, an enormous crew of workers.
Sounds like a dream project, really.
Right. But there's this fatal flaw in your operation. Your construction workers, they only know how to manufacture straight pipes. I mean, they possess absolutely no idea how to branch them or angle them or, actually connect them to individual buildings.
Oh, wow. Yeah, that's a problem.
Exactly. If you let them loose, you wouldn't get a functional city grid. You would just get this massive, chaotic mountain of PVC.
Which is basically a logistical nightmare, because you have this abundance of raw material, but absolutely zero functional architecture. I mean, in any fluid dynamics system, if you lack directed branching, the entire network just completely fails.
And that precise scenario, well, that brings us to one of the most fascinating biological mysteries in recent literature. I mean, how does the human body know how to branch its internal plumbing? Specifically, its lymphatic vessels.
Right, the lymphatic.
Yeah. So today, we're doing a deep dive into this brilliant 2026 Nature Communications paper by Schoofs and an international team of researchers. The mission of this deep dive for you listening today is to unpack the exact granular mechanisms of how lymphatic vessels grow a process called lymphangiogenesis, and to finally resolve this really contentious scientific debate about two cellular antennas, which are known as VEGFR2 and VEGFR3. So, okay, let's unpack this.
We really need to, because the stakes here, they go far beyond just academic curiosity. We are looking at the foundational mechanics of tissue homeostasis.
Yeah.
If we can map out the precise biomolecular signaling that dictates how these vessels branch, I mean, we hold the potential keys to controlling chronic inflammation, mitigating severe lymphedema, and critically starving metastatic cancer cells of their primary escape routes.
Which is huge. But before we can understand the branching, we really need to map out the signals for you. So let's set the stage. The lymphatic system, it operates as the body's secondary fluid highway, right?
Right. It's totally separate from the blood.
Right. Exactly. Unlike blood vessels that form this continuous pressurized loop, lymphatic vessels are blind-ended. They function as a highly specialized cellular vacuum network. They absorb interstitial fluid, macromolecules, immune cells from your tissues, and funnel them all back into the bloodstream.
And if that vacuum system fails or if it develops improperly, the result is localized fluid retention and just massive tissue swelling.
Wow.
Yeah, it's severe. So to construct these intricate networks, the body relies on a very specific set of molecular players. The main one, the master signaling protein driving this growth, is VEGF-C.
Okay, VEGF-C.
Right. And you can conceptualize VEGF-C as this highly specific physical key drifting through the extracellular space.
And obviously, for a key to do anything, it needs to find a corresponding lock on the surface of a cell.
Precisely. And the primary lock, like the main receptor on the surface of these lymphatic endothelial cells, is VEGFR3. Whenever scientists analyzed lymphangiogenesis in the past, VEGFR3 was universally acknowledged as the undisputed star of the show. I mean, when the VEGF-C key inserts into the VEGFR3 lock, it triggers this intracellular cascade that just gets the cells dividing and moving.
But the paper focuses really heavily on the mysterious second lock, which is VEGFR2. And here's where I have to push back a bit, because VEGFR2 is famous, but not for lymphatics, right?
Not at all.
It's the master builder for angiogenesis, the growth of blood vessels. So when it comes to the lymphatic system, the historical consensus seemed to be that VEGFR2 was practically useless, like maybe just some redundant evolutionary hangover.
Exactly.
Why would the body even bother expressing this powerful blood vessel builder on a lymphatic cell if it isn't going to use it?
Well, that perceived redundancy, that's the exact paradox that launched this entire investigation. The researchers were looking at this glaring contradiction in the literature.
Which is what?
So when scientists studied these lymphatic endothelial cells in vitro, isolating them in a culture dish, VEGFR2 was remarkably active. Oh, really?
Yeah, it was driving heavy cellular reactions, promoting cell survival, initiating migration. But, and here's the kicker, when they moved to in vivo models, specifically genetically modifying living mice to completely lack the VEGFR2 gene in their lymphatic system, nothing happened.
Wait, nothing?
Nothing. The mice developed perfectly normal functional lymphatic networks.
That seems entirely contradictory. I mean, the receptor is highly reactive when it's isolated in a dish, but completely silent in the actual living organism.
It totally defied logic. The entire scientific community had effectively built a consensus around those in vivo knockout studies, and they concluded that VEGFR2 was just a non-essential player in lymphatics.
Wow.
But the team behind this 2026 paper, they hypothesized that the older models were fundamentally flawed, and they set out to prove it by dissecting the signaling pathways with just unprecedented precision.
Okay, let's synthesize what we have so far, because this paradox is really the foundation of the whole mystery here. So if you're tracking the core variables at home, we have VEGF-C acting as the master key.
We have VEGFR3 acting as the primary lock that drives cell activity, and we have VEGFR2, this receptor that conventional wisdom dismissed as completely useless because knocking it out in living animals didn't disrupt the system at all.
Exactly.
So I want to pose a question to you, the listener. Put yourself in the shoes of these researchers. How do you prove a receptor is critical when the entire biological system appears to operate perfectly fine without it?
Well, you have to eliminate the noise. You can't just look at the whole system. You have to isolate the individual signaling pathways to see what each receptor does in a total vacuum.
Right.
And to accomplish this, the research team utilized adeno-associated viral vectors, or AAVs.
I really want to dig into that methodology, actually, because AAVs are fascinating. They're essentially stripped-down viruses, right? Like, the researchers remove the viral replication genes and replace them with a specific genetic payload.
That is exactly the mechanism. They engineered these viral vectors to express very specific mutated versions of the VEGF signaling proteins, and they injected these AAVs into the ear skin of mice.
Why the ear?
Because it's an ideal, highly vascularized, yet incredibly thin tissue layer. It's just perfect for high-resolution imaging.
Oh, that makes sense.
Yeah, and this allowed them to run 3 distinct tests. In the first test, they delivered a viral payload called AAV-VEGF-A164 that binds exclusively to VEGFR2.
Okay, so they stimulated the supposedly useless receptor all by itself. What did it actually do?
It triggered severe hypertrophy. The lymphatic vessels became massive, enlarged, and incredibly thick. But, and this is crucial, there was absolutely no sprouting.
Wow.
0 new branches formed. It just created these giant straight pipes.
Which proves it isn't silent at all. It's doing something very specific, reinforcing the structure. So what happened in the second test when they isolated the star of the show, VEGFR3?
For the second test, they utilized a mutated signal that only binds to that main receptor, VEGFR3, and the physiological response was drastic. The tissue developed severe lymphatic hyperplasia.
Meaning what exactly?
The endothelial cells just entered this state of rapid, chaotic proliferation. They multiplied endlessly, creating these dense, completely disorganized cellular mats. But just like the first test, there was no functional directed branching.
Here's where it gets really interesting. By isolating these pathways so cleanly, they just shattered the existing paradigm. The researchers proved that neither receptor is capable of building a functional network autonomously, right?
Exactly.
If you only activate VEGFR3, you get a chaotic pile of rapidly multiplying cells, basically our mountain of PDC pipes from the intro.
Yeah, a useless mountain.
And if you only activate VEGFR2, you just thicken the existing walls without making any new turns.
And that leads directly to their third test. They introduced the native wild-type VEGF-C protein, the unmutated signal that simultaneously binds to both VEGFR2 and VEGFR3 at the same time.
And the result?
A beautifully organized, complex, and highly branched functional lymphatic capillary network.
Incredible. So to actually build the network, you definitively need both of them working together.
To absolutely do.
Let's do a quick 10-minute recap here just to frame this up for you and keep these mental models sharp. When the researchers isolated the signals, they found that VEGFR3 strictly drives cell proliferation, while VEGFR2 strictly drives vessel enlargement. But functional branching, the actual architectural design of the network, only occurs when both receptors are activated simultaneously.
Right.
Think about it like a high-stakes engineering project. Yep. If one team only procures raw steel and the other team only pours concrete, who's actually looking at the structural blueprints to build the bridge, they have to coordinate.
It's all about coordination.
But wait, this leaves a massive glaring contradiction.
The in vivo knockout studies.
Yes. I mean, if this 2026 paper just definitively proved that both receptors are absolutely mandatory for branching, why did decades of prior genetic knockout studies show that deleting VEGFR2 in a living mouse did absolutely nothing?
Okay, this is perhaps the most elegant part of the entire paper. It exposes this fundamental flaw in how we perform conditional genetics. And it revolves around a phenomenon known as escaper cells.
Escaper cell.
Yeah. When geneticists want to study gene function, they typically use a system called Krelok's recombination to effectively scissor a specific gene out of the DNA.
But those biological scissors aren't perfect, are they?
Far from it. The standard Krelok system is highly stochastic. In a best-case scenario, it might successfully delete the target gene in maybe 85 or 90% of the cells within a tissue.
Okay.
That leaves 10 to 15% of the cells entirely unaltered. In older studies, researchers just assumed that a 90% deletion rate was more than enough to observe a functional deficit in the tissue.
But they vastly underestimated the competitive nature of cellular biology, didn't they? Those 10% of cells basically escaped the genetic deletion. They still possessed their functional VEGF-R2 receptors.
Yes, and those escaper cells weaponized that advantage. Because they possessed the complete molecular toolkit required for branching, while their neighbors did not, the escaper cells rapidly outcompeted the deficient cells. They aggressively migrated to the leading edge of the growing vessels, performed all of the active sprouting, and single-handedly constructed the new vascular network. They effectively masked the genetic defect of the surrounding 90%.
That is just brilliant and incredibly deceptive. Think about trying to evaluate if a runner's performance is hindered by wearing heavy combat boots.
Okay.
You outfit a whole relay team with the boots, but one runner secretly sneaks onto the track wearing professional carbon fiber racing cleats. That's our escaper cell.
I love that analogy.
Right. That single runner is going to dominate the race, carry the entire team to the finish line, and you will look at the final stopwatch and falsely conclude, well, the data shows heavy boots don't slow you down at all.
The entire scientific consensus was built on cells essentially cheating the assay.
Exactly. And to bypass this biological cheating, the 2026 team deployed an advanced, high-fidelity genetic tool called the ICER-Cre system.
How is that different? Well, standard Cre systems rely on probabilistic, independent events. The ICER-Cre system fundamentally alters that dynamic. It engineers this strict dependency where the expression of a fluorescent reporter protein is biologically tethered to the absolute certainty of the gene actually being cut out.
Meaning, if the cell glows under the microscope, the researchers know with 100% molecular certainty that the EGFR2 gene has been completely obliterated. There's no guesswork.
Exactly. It guarantees complete penetrance. And when they utilized ICER-Cre to unconditionally eradicate VEGFR2 from every single lymphatic endothelial cell, lymphangiogenesis suffered a catastrophic failure.
Total collapse.
Total collapse.
Yeah.
The lymphatic capillaries were utterly incapable of forming even a single sprout. The architecture just failed entirely.
Which completely rewrites the textbook. VEGFR2 is not a redundant backup. It is a strictly mandatory structural component.
It absolutely is.
So let's pause and do our 15-minute recap to digest this massive pivot. The older accepted scientific facts were essentially an illusion created by a small minority of hyper-competitive escaper cells that masked the absence of VEGFR2.
Right.
And when those cells were strictly controlled for, using the new tools, the absence of VEGFR2 completely paralyzed the system's ability to branch.
Yeah.
It raises this really fascinating and honestly somewhat terrifying question for you to ponder. How many other established biological paradigms are currently resting on the deceptive shoulders of cells that simply sneak past our genetic scissors?
It is a profound methodological warning for the entire field of genetics, honestly. But establishing that both receptors are mandatory only solves half the mystery.
Right, the what?
Exactly. We now have to address the mechanism, the how. How do these two distinct receptors physically orchestrate this branching behavior on the cellular membrane?
Okay, we need to dive into the biochemistry. The paper outlines a very specific sequence of events, so let's use a mental framework for you listening to track this. Bind, push, lock.
That maps perfectly to the biomolecular cascade. So, step 1, bind. The wild-type VEGF-C protein arrives in the extracellular space and binds to VEGFR3. Now, VEGFR3 has a naturally higher binding affinity for it than VEGFR2 does.
Okay, so it grabs it first.
Right. And upon binding, VEGFR3 initiates that chaotic cell proliferation we saw earlier. But it also rapidly internalizes, meaning it is physically pulled from the surface into the interior of the cell.
Okay, so that initial binding is the catalyst. Now we enter step 2 of our framework. Push.
Yes. The activation of VEGFR3 triggers an intracellular signaling cascade that heavily involves a specific lipid kinase known as PI3K alpha.
Okay, let's conceptualize PI3K alpha as the pusher. But how is it physically pushing anything around inside the cell?
Well, PI3K alpha functions by altering the actual lipid composition of the cellular membrane. It phosphorylates a lipid called PIP2 and turns it into PIP3. This localized change in the membrane chemistry acts as a highly specific docking signal. It essentially recruits intracellular vesicles. You can think of them as tiny biological cargo pods that are loaded with dormant VEGFR2 proteins.
Oh, wow.
Yeah. The modified membrane essentially pulls these cargo pods to the surface, causing them to fuse with the outer membrane. So it's pushing the hidden VEGFR2 receptors out from the inside of the cell into the open environment.
That is wild. The primary lock receives the key, which triggers the pusher to alter the membrane, which literally shoves the secondary locks up to the surface.
Exactly.
And that brings us to step 3, lock.
Right. So once VEGFR2 is localized to the surface membrane, it encounters the remaining VEGFR3 receptors. And driven by the presence of that VEGF-C protein, the two distinct receptors physically bind to one another.
Wait, they physically connect?
Yes. They form what's called a heterodimer. It's a massive combined superreceptor complex.
The heterodimer. I mean, it sounds incredibly dramatic, like a sci-fi movie, but it really is the molecular climax of this entire branching process.
It really is, because it fundamentally rewrites the cell's internal programming. Think about it. When VEGFR3 was acting alone, it broadcasted this localized signal telling the cell to just endlessly divide.
Right. The PVC mountain.
But the moment VEGFR2 locks into it, forming the heterodimer, the actual shape, the conformation of the intracellular domains shifts entirely.
Wow.
The signaling cascade alters drastically. The cell stops blindly multiplying, it reorganizes its cytoskeleton, and it begins directed functional sprouting.
The sheer elegance of that is just stunning. The introduction of the key doesn't just activate the lock, it changes the shape of the locks, forcing them to combine, which changes the entire behavioral output of the cell.
It's a beautiful system.
But I have to ask about the burden of proof here. Because cells are microscopic, receptors are nanoscopic. How on earth did the researchers definitively prove that these two receptors are physically locking together on the surface of a living cell?
The methodological rigor here is honestly what got the paper into Nature Communications. They utilized a technique called a proximity ligation assay, or PLA.
Wait, what's a PLA?
PLA is essentially a molecular proximity alarm. You introduce two distinct antibodies into the tissue, one engineered to bind to VEGFR2, the other to VEGFR3. But crucially, attached to the tail of each antibody is a tiny, distinct strand of DNA.
Okay, so you have these little DNA strands just dangling off the receptors.
Right, and those DNA strands, they can only interact if they are brought within 40 nanometers of each other.
Wow.
If the two receptors lock together, the DNA strands are close enough to be enzymatically ligated, meaning joined together to form a complete circle.
Okay.
The researchers then introduce an enzyme that performs rolling circle amplification. It takes that tiny circular DNA and multiplies it thousands of times until it forms this massive bundle that brilliantly fluoresces under a microscope.
Okay, 40 nanometers, just to put that into perspective for you. A single sheet of paper is about 100,000 nanometers thick.
Yeah.
So if these receptors are separated by even a fraction of that paper's thickness, the test remains completely dark.
Exactly. But in the living tissues that were exposed to wild-type VEGF-C, the PLA assays lit up with incredible intensity. The fluorescent signals provided irrefutable physical proof that VEGFR2 and VEGFR3 were actively tethered together, forming heterodimers on the actual cell surfaces in vivo.
That biochemical sequence is just beautiful. But I want to ground this in practical reality, if I suffer a laceration, why does this heterodimer mechanism matter?
The researchers didn't just study pristine tissues, right? They utilized a regenerative wound healing model.
Yes, specifically an ear punch biopsy model.
Okay.
When tissue is damaged, the lymphatic network is severed. To restore fluid homeostasis and allow immune trafficking, the body has to regenerate those vessels across the empty wound cap.
Right.
When the researchers induced these biopsies in mice lacking the main receptor, VEGFR3, the regenerative process was completely paralyzed at phase 1.
Yeah.
The cells simply couldn't multiply.
But the critical test is what happens when they utilize that Eicher-Cree system to exclusively delete the secondary receptor, VEGFR2, during wound healing. What happened then? The resulting pathology perfectly validated the bind, push, lock mechanism. Without VEGFR2, the endothelial cells could still multiply because they were driven by the remaining VEGFR3. But they completely lost the ability to form the highly specialized, spiky tip cells required to actually physically bridge the gap. The proliferating cells just amassed at the edge of the wound in these disorganized clusters. They were entirely incapable of directional sprouting.
Let's bring all of these complex mechanics together for you for our final recap, because the sequence is vital to understand the ultimate medical application here.
Definitely.
We've established that the process requires a synchronized molecular dance. Blind, push, lock. VEGF-C binds to the primary receptor, triggering cellular division. The lipid kinase PI3K alpha alters the membrane chemistry, pushing the secondary receptor, VEGFR2, to the surface. Finally, the two receptors physically tether together, forming a heterodimer complex that shifts the cellular program from chaotic multiplication to targeted architectural branching.
Perfectly summarized.
So consider the implications of that wound healing model for a second. If you are trying to regenerate tissue across a massive gap, do you want to flood the system with drugs that merely force cells to divide? Or do you need a targeted therapy that ensures they can actually sprout and connect the plumbing?
You inherently require both.
Yeah.
And that realization forces a massive paradigm shift in how we approach targeted therapeutics.
So what does this all mean for the future of medicine?
Well, it implies that our entire pharmacological approach to the lymphatic system has been, honestly, fundamentally myopic.
How so?
For years, therapeutic development for lymphatic disorders has almost exclusively focused on agonizing or antagonizing VEGFR3 alone.
Right.
But if your goal is to achieve optimal modulation of lymphangiogenesis, whether you're attempting to promote it to heal a severe wound or actively inhibit it to prevent a tumor from establishing a lymphatic escape route for metastasis, targeting a single receptor is mathematically insufficient. You must engage the entire heterodimer complex.
Because, if you only block VEGFR3, the remaining VEGFR2 might be recruited by alternate pathways, or vice versa, leading to completely unpredictable clinical outcomes.
Precisely. But the most exciting translational revelation in this paper involves existing pharmacology.
Ooh, do tell.
The pharmaceutical industry has spent decades developing highly potent drugs designed to inhibit VEGFR2. We currently utilize these anti-angiogenic therapies in oncology to starve blood vessel formation in tumors.
Oh, but because the historical consensus falsely labeled VEGFR2 as useless in lymphatic tissue, no one bothered to apply those existing drugs to lymphatic pathologies.
Exactly. This research suggests a massive opportunity for drug repurposing.
That is incredible.
For patients suffering from severe, uncontrolled lymphangiogenesis, like specific chronic inflammatory conditions, persistent lymphedema, or even corneal transplant rejections, where invasive lymphatic vessels literally destroy tissue viability, we might not need to develop novel compounds from scratch.
The drugs are already there.
The precise molecular tools to shut down that branching might already be sitting on the pharmacy shelf, heavily tested and FDA approved. We simply lacked the accurate biological blueprint to understand where to deploy them.
That is the epitome of transformative science. Today, we dissected how a cellular receptor long dismissed by the academic community as a useless relic quietly sitting on the sidelines was actually a critical, dormant architect. It was merely waiting for the precise molecular sequence to lock arms with its partner and fundamentally rewrite the behavior of the cell.
It's an amazing story.
It really is. And it forces you to look at the vast complexity of the human genome through a completely different lens. It leaves you with this thought. What other seemingly redundant or useless genes lying dormant in our DNA are simply waiting for the right molecular chord to be struck before they reveal their true magnificent purpose? Thank you for joining us on this deep dive. Keep questioning the invisible blueprints of the world around you.